Hi there. I'm Priscilla Hsue. I'm president of the American Society of Clinical Investigation. I'm so honored to speak today with Dr. Joe Wu, who's this year's recipient of the Stanley Korsmeyer Award. This award recognizes outstanding scientific achievement to advance knowledge in a specific field and in mentoring future generations of life science researchers. Dr. Wu is director of the Stanford Cardiovascular Institute and also the Simon Stertzer Professor of Medicine and Radiology. Um, Dr. Wu, could you please tell us a little bit about yourself, your training path, and how you came to study the problems you're currently working on in your lab?
Oh, Priscilla, thank you so much for inviting me to be here, to be the recipient of this year's Korsmeyer Award. And I've been an ASCI member for a long time, and I think this is probably the greatest organization to promote physician scientists. And also, it's a great honor to receive this award, given how influential Dr. Stanley Korsmeyer was in terms of being a champion for the cause of the physician scientists. So I did my undergrad at UCLA, and after undergrad, this is back in '93, I went to uh, Yale for medical school, and at that time, I wasn't sure if I want to do MD or versus MD PhD. I was actually accepted to the MD PhD program at UCLA, but I didn't have the confidence that I could be a physician scientist, so I just went to MD program instead to Yale. Luckily, at Yale, they had a great program in the sense that during medical school, you had to do a thesis, a research thesis. At the same time, they didn't emphasize grades that much, and in fact, I still remember that when you take an exam, you don't have to put down your actual name.
Oh, wow.
And so my name, every time when I took an exam, I actually put down Joe Montana. It's a fake name. It's an honor system. If I fail and it gets posted, Joe Montana, score is 55, cutoff is say 70, then it's an honor system I need to report to the teacher.
Wow.
But because of the system, it de-emphasized competition among medical students, and it emphasized doing some type of research. And so I did my research with Dr. Hsue. At that time, I was interested in being a cardiothoracic surgeon. And so I did a lot of canine research, trying to understand the coronary flow dynamics. And then after, I would say, 4 straight summers, I kind of tap out and say being a cardiothoracic surgeon is very difficult
Yep.
if you want to do research. And so when I applied to UCLA for the internship and residency program, I specifically applied to the UCLA STAR program.
Mhm.
At that time, as I mentioned earlier, I didn't go to the MD PhD program at UCLA. I chose the Yale program. But then I realized I really enjoy research at Yale because it gave me confidence that I can actually do it. I published 2 papers as a medical student, even though they won the highest impact factor journal, but it actually gave me a lot of joy to get it published. And so I went back to UCLA for my internship, residency, cardiology fellowship, and I did the STAR program. And I think the STAR program was quite instrumental in the sense that it gave you 4 years of protected time for you to do research. And I think at that time, the program director was Dr. Joy Frank, and then followed by Dr. Linda Deemer.
Yep.
And and then Dr. Alan Fogelman set up the program, and then also Dr. Jim Weiss was the chief of cardiology. I would say everybody at UCLA, they were very, very encouraging of us cardiology fellows, or just fellows in general, to go into the program. They give you a lot of guidance, they give you a lot of mentorship, and they really believe in the fellows. And I think during that period of time, I also got quite lucky in the sense that I picked Dr. Sam Gambier as my PhD advisor. And so Sam and I learned quite a bit from Dr. Gambier during the 4 years in his lab, and we were working on some of the cutting edge technologies of how do you do molecular imaging to track stem cells, to track gene therapy. And because of my experience in his lab, I felt like finally, I can kind of cut it as a physician scientist. So it's long-winded way to answer your question, but I must say that along the way, there were a lot of influential figures who guided my decision to become a physician scientist. Yeah.
Wonderful. Thank you. Um, how did you first become interested in using stem cell models for cardiovascular disease?
Yeah. So my PhD was mostly on molecular imaging, and molecular imaging just means that instead of using a traditional immunofluorescence in which you have to quote unquote sacrifice the animal to stain the tissue to see where your gene expression is, where your stem cells are. Uh, Dr. Gambier and his colleagues developed techniques in which you could image and see where the stem cells are or where the gene therapy is. He was interested in mostly cancer stem cells, whereas I went to his lab and I said, hey, can I use the same technique and track what happened to the cardiovascular stem cells? And so what happened was I worked in his lab, and during my PhD, we were working mostly with adult stem cells, with mesenchymal stem cells, with bone marrow stem cells. And also we were working quite a bit with cardiac gene therapy, because at that time it was quite quote unquote hot. People were using adenovirus to overexpress VEGF, vascular endothelial growth factor, to see if it could induce um, angiogenesis in the heart. But what happened was that when we injected these bone marrow cells, the mesenchymal stem cells, or the adenovirus with VEGF into the heart, by imaging, we could track within the same animal that most of the stem cells were gone in about 2 weeks. And likewise, by imaging, we could track within the same animal that most of the adenoviral VEGF gene expression was 2 weeks. And I was doing my PhD in pharmacology. And in pharmacology, the first thing they teach you is PKPD, right? Pharmacokinetics, pharmacodynamics.
Yep.
And I realized, wait, that doesn't probably make sense, because if the cells are gone in 2 weeks, if the genes are gone in 2 weeks, then it's probably very difficult to get a doable angiogenesis or doable engraftment. And so when I went to Stanford in 2004, at that time, then we started playing with other cell types. Other cell types, specifically meaning human embryonic stem cells.
Right.
And with human embryonic stem cells back then, there was a ban on using human ES cells by President George Bush. But because I had a starter package from Stanford, and the starter package was not a federal money, so it wasn't encumbered
I
by
see.
the ban.
Right.
And I could still remember that when you buy a equipment, you have to document whether it was NIH funded or not. Because I didn't have NIH funding, I could buy any equipment I want that's non-NIH funded. So we actually started working with the human embryonic stem cells. We work with a couple labs. For example, Dr. Julie Baker's lab at Stanford. We learned from her lab, how do you grow human embryonic stem cells? How do you track human embryonic stem cells? And I still remember, one of the early experiments that we did was we labeled the human embryonic stem cell with firefly luciferase, and also with GFP, and also with HSV-TK, something called triple fusion reporter gene, and we injected into the heart, and the signal took off. Basically, at 2 weeks, 3 weeks, 4 weeks, the signal went up very, very high. I never seen those signals because, as I mentioned earlier, what our experience of bone marrow cell mesenchymal stem cell was that most of the signals are gone, disappear by about 2 to 3 weeks, and the signal took off. And when we sacrificed the animal at that time, it actually turned out to be a teratoma. Teratoma just means that the human ear cell, because it was unguided, it became all the cell types within the body. So you could actually identify bone cells, brain cells, liver cells, kidney cells, heart cells. So it was disappointing, but at the same time, it also opened up my eyes and say, whoa, this is a cell type that can become any cell types in your body, and therefore you should be able to take human embryonic stem cells and add different growth factors, different cytokines, different cocktails, so that you could push the human embryonic stem cell into brain cell, into heart cells, into liver cells, into kidney cells. And so we spent, for the next 2 years, we spent a lot of effort between 2004, 2006 to take a human embryonic stem cells and how to push it into beating cardiomyocytes. And then around 2006, Dr. Shinya Yamanaka published a paper in Cell showing that you could actually take a mouse fibroblast, add 4 genes into it, and it will become a mouse iPS cell, so induced pluripotent stem cell. And these iPS cells, for all practical purposes, are very similar to the mouse ES cell. And then you can then take these mouse iPS cells and become all the different cell types. And he followed that up in 2007 to show that you could do the same thing in human iPS cell, meaning that you could take a human skin fibroblast, add the 4 genes, and make it human iPS cell, and then differentiate all the cell types. And I still remember that, at that time, it kind of dawned upon me that as a clinician, as a physician scientist, the main difference between human ear cell and human iPS cell is that for human ear cell, I never know who the donor was
Mhm.
because it's a quote unquote destroyed embryo.
Right.
Whereas with human iPS cells, I know exactly who that patient is, and I could follow that patient over time, and I can use that patient's clinical history to correlate it with what happened to the human iPS cell. It's almost like what we call today the biological twin. Essentially, I'm creating a biological twin,
Right.
and so we decided to pivot from human ESL all in with the human iPSL. And luckily, we had some early grant fundings from the California Institute of Regenerative Medicine.
Yep.
And then with the human iPSLs, and it's really up to your imagination to see what is it that we need to add to push the human iPS cells into cardiomyocytes, to push it into fibroblasts, to push it into coronary endothelial cells, to push it into parasites, to push it into smooth muscle cells, what I call the big five cell types within the heart, as well as other cell types such as macrophage. And so we spend a lot of time developing these protocols so that we can reliably and reproducibly generate these cells. And then once you're able to generate human cardiomyocytes from healthy versus patients with long QT syndrome versus patients with hypertrophic cardiomyopathy, dilated cardiomyopathy, ARVC, then again, it's really up to your imagination to see how are you going to use this for disease modeling. Yeah,
Got it.
yeah.
Wonderful. Thank you.
Mm.
So, Joe, I gotta say, as I'm a little shocked to hear you say the beginning, early in your career, you did not feel confident to pursue physician scientists. Um, what factors do you feel have been very critical to your success in the lab?
Mhm. Yeah, so I come from a humble background. I mean, I'm the first. I guess my brother and I were the first kids in the extended family to go to college, and I'm definitely the first one to go to medical school, right? So it's not like I come from a long, tradition of a family of doctors and so forth. So I didn't really have confidence in the beginning that I can really cut it, um, but then, as I mentioned earlier, that I got lucky in the sense that at Yale, they emphasize research. At UCLA, the STAR program, we also emphasize research and provide great mentorship. And so, and then I was lucky to to be mentored by Dr. Sam Gambier, who unfortunately passed away. But I would say, the main thing that's important for the success of any physician scientist, any trainees, any PhD scientists and clinician scientists is really mentorship, right? And so finding the right mentor can really make a big difference. And I think there are a lot of talks about, finding resiliency and having the right mentor, having the right environment for your research. And certainly Stanford and UCLA, they're great environments for research. And that's part of the reason why I've been able to succeed at Stanford. People talk about networking and people talk about, having grit and so forth. I try to kind of boil down to something very simple for our fellows, cardiology fellows, or for our postdoc trainees. And because I know all of them will probably think in the same way as I was thinking 25 years ago. That is, if I set up a lab or if I go into research, can I do exactly the same way as this big PI or big physician scientist is doing? So I actually boil it down and make it very simple. I mean, I would sit down the fellow, the postdoc, and I would say, pretend it's John. I would say, hey, John, you want to become a physician scientist, right? And I just say, it's very simple, very, very simple. And I just say, number one, are you okay with the pay cut? And I say you have to be okay with that because you're not working as hard as our clinicians who are doing full time.
Right.
And so you have to be okay with the slight pay cut. And John would say, yeah, I'm okay with the pay cut. And number two, I would ask, is the spouse okay with the pay cut?
Right.
Right? And these are very important questions because I do think that a lot of the young MD, PhD students, they don't understand these practical questions that I'm asking.
Yep.
And if John says, yeah, I think my girlfriend or my, current wife is okay. And I say, number three, are you willing to come in on the weekends writing grants and papers?
Yep.
Right? Because as a physician scientist, everybody has 24 hours in a day, and so you got to squeeze in time somewhere to come in to write grants and papers. And I think if the answer is yes, 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 then I'd tell him or her that number four, find a great mentor to help guide you, to show you how it's done. And after that, it's not that hard. It is not that hard. And so that's why I try to de-escalate the fear and de-risk everything for the trainee and say, if you say yes, 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 and find a great mentor, it is not that hard to stay in this game to be a physician scientist. Yeah.
Good advice. Um, how has the preclinical drug discovery process evolved with the integration of human relevant in vitro models
Mhm.
and AI approaches, and kind of what do you see on the horizon?
Yeah, so I'm a believer that you want to integrate and you want to take advantage of all the new tools that are available to advance the science, right? Because at the end of the day, you want to come up with something that you want to cure and treat and help patients. And so a lot of these tools that are present, um, they were not present when I was doing my PhD, right? And certainly, for example, during my Korsmeyer lecture, I mentioned that in our lab, we do a lot of next generation sequencing, right? All the sequencing techniques, the single cell RNA sequencing, the single cell ATAC-seq, those were not available back around 2000, 2004 when I was doing my PhD. And number two, I talk about CRISPR editing. Well, that came around in 2010, right? Those were not available. And I also talk about the iPSL technology that came about in 2006, 2007. Again, those were not available during my PhD. And lastly, I talk about the AI and how I mentioned that individually, these are all very important, powerful techniques, but individually, they're not able to help you address the question of drug discovery. And so you need to combine all four of them to help you accelerate that steps. So if you take a look at the advances that have occurred in the past 20 years, and if you're able to say, for all diseases, if I want to come up with the AI-based drug discovery, for all diseases, I need to start with the disease.
Mhm.
I'm going backward, right? So most of the time, people want to have a technique, and then with the technique, come up with a cure for disease. That's what I call A to Z.
Mhm.
But I'm going backward. I'm going from Z to A.
Right.
Z means I need to start with the disease. What is the unmet need about the disease? Why is it that some treatments work and some treatments don't work? What is the mechanism of the disease?
Mhm.
And then what is the target for that disease? And once I identify the target, then I can use, for example, AI softwares to figure out what the protein structure is, for
Mhm.
example, the AlphaFold, and also some of the X-ray crystallography structures. I can then use AI softwares to help me do predictions of what drugs dock to that particular protein domain.
Mhm.
And then once I synthesize these drugs, I can then take these drugs and test it on patient-specific organoids or
Mhm.
patient-specific stem cells that have the disease phenotype, right? If I want to come up with a drug for Brugada syndrome, a drug for dilated cardiomyopathy, I want to test it on human iPSL-derived cardio organoids that has a dilated cardiomyopathy that has the Brugada syndrome rather than a mouse model because its phenotype is very different. And then I can then see what drugs work and what drug doesn't work. And by integrating functional genomics, meaning that if I want to confirm that the target is the culprit, I can then do CRISPR editing on these cells
Mhm.
and to knock in and knock out the particular gene to show the disease phenotype goes away. And then I can do it on a large scale because I can go to my clinical trial in a dish model by recruiting a lot of Brugada patients or a lot of dilated cardiomyopathy patients, have these cells on a dish, test how the drug work and why the drug doesn't work. So you could do a lot of that,
Mhm.
this type of research right now for drug discovery just by using the four models I mentioned, the next-gen sequencing, the CRISPR editing, the iPS cells, and the AI-based platform. Yeah,
I see. Wonderful. So the next question is about mentoring
yeah.
and how you've approached mentorship in your lab and in your role as institute director.
Yeah, so I think as an institute director, uh, I interact and with many postdocs, many graduate students, and also many young and middle career
Mhm.
faculty. Right, I think for mentorship, we are in this game or business or this field, academia. In my opinion, I think the biggest reason we're in it is really to help train the next generation of
Mhm.
scientists, next generation clinicians, and next generation physician scientists. And so it serves you no good if we don't have uh, the next set of people taking the baton from you, because I'm not going to be in this field for next 50 years. So at some time I got to retire in the next 10, 15 years and pass it off to the next generation. So it is very important for me personally, and it's also very important that for our Stanford Cardiovascular Institute, we have a great infrastructure set up. For the Cardiovascular Institute, for example, we have 6 different T32 training grants,
Wow.
looking at imaging, looking at vascular biology, myocardial biology, looking at computational science, looking at epidemiology. We also have a R38 grant to train the physician scientists.
Mhm.
We also have a R25 and a T35 grant,
Mm.
in which each year we bring in about 40 to 50 undergrads throughout the country who are not traditionally exposed to research and come to Stanford, work with different faculty within the Cardiovascular Institute. within that 10 weeks, learn how research is done. They can then go back to their home institution and then apply to MD or apply to a PhD program.
I see.
So I, as the institute director, I write a lot of letters
Right.
for people who come to this summer program, and they then apply to these graduate school, and a lot of times they'll email me back and say, "Hey, dear Dr. Wu, I got into this medical school or
Mm,
that medical school." So
that's
it gives
great.
you a lot of satisfaction to see that the trainees who come through this program get into it. I think on a individual lab level, I also have a lot of place a lot of emphasis that my trainees do well. So, for example, the trainees who come to my lab, I would teach them how do you write a grant, how do you review a grant,
Mm-hmm.
how do you network, and how do you, um, you know, understand what are the most important problems in research and be able to have a keen acumen and say that this is important, this is not important. And so luckily, I think over the past 20 years, we've had 62 by my last count,
Wow.
62 trainees who've set up PI, who've become PIs to set up their own
Wow.
labs. I think last year, for example, we had postdocs who went to Johns Hopkins in the biomedical engineering department, and then another postdoc went to Hsue.
Mm-hmm.
Another postdoc went to UT Southwestern.
Okay.
Another postdoc went to The Ohio State University.
Mm-hmm.
Another postdoc went to University of Montreal, and then another postdoc went to University of Galway in Ireland. And then this year, we have several postdocs who are on, it's almost like a going to the airport. It sounds almost like airplanes taxiing, ready to take off.
I
So as
see.
you know, we had recently last month, we have Hsue who joined
Yep.
UCLA Cardiovascular
Yep.
as a assistant professor. All of these give me a lot of joy to see them succeeding. In fact, last week, I took one of my former postdoc, Dr. Sayed, who's now associate professor at Stanford, to dinner with his family and two kids, and to celebrate him receiving the R35 Outstanding
Mm,
Investigator Award, right? And so it's just a
wonderful.
great joy to see the next generation of trainees doing so well successfully, and then able to take science to the next step, because ultimately, as I mentioned earlier, at the end of the day, all of us want to improve human health. And these are the folks who can help me and who help everybody to take it to the next step. Yeah.
Well, you kind of touched upon this already, but the last question is how to best encourage and support young physician scientists in their career.
Yeah, so I think for the young physician scientists, I think the mentorship is the key, right? And I try to, again, I try to tell the young physician scientists that it is really not that hard if you follow those four principles. Start with physician scientists, especially physician scientists, start with the concept that you're gonna have to willing to sacrifice to take a small pay cut. I don't think a lot of people discuss this
Right.
at all. And I
It's
do
true.
think that for the young trainees, they have to understand that, because you don't want to go through 8 years of MD, PhD, another 3 years or 5 or 6 years of clinical training. In the end, realize that you're not willing to accept that pay
Right.
cut. And in my opinion, I think that pay cut is actually reasonable because, as I mentioned earlier, I work with our clinicians, and they're working very, very hard.
Yes.
They don't have the luxury as many physician scientists who can travel and give talks and go to meetings, set their schedule. So I mentioned earlier that pay cut, spouse okay with the pay cut, working hard on the weekends, because running a lab is almost like running a business, right? And then also finding the right mentor. Separately, I also kind of advise them that especially during their training phase, I think they just need to learn the three principles of, at least in my lab, I kind of uh, teach my trainees that they have to work hard, right? Because you can't succeed if you're not working hard. And number two, they got to work smart,
Right.
meaning that uh, if there are new technologies, incorporate them. If the fundamental hypothesis is incorrect, then you got to be able to be nimble enough to pivot to another angle, to another direction. And then number three, I tell them that you had to work collaboratively. And I always ask them which one of the three is the most important.
Uh-huh.
And sometimes they kind of guess one or the other, but I also emphasize to them that among these three, the most important part is working together,
Yes.
because you can only go so far by working hard. You can only go so far by working smart. But if you work together as a team, you can go much, much, much farther. And I hope all my trainees understand that, and I do hope that for the young physician scientists who are listening to this conversation, that they understand that working together as a team, because science is a team sport. It is the most important thing, being collaborative, being collegial, willing to share material, sharing ideas, because as I mentioned, ultimately, you're not doing it for your own sake. You're doing it because you want to advance science to help out patients.
Wonderful. Well, thank you so much for
Thank you.
taking the time to talk to me today, and a very big congratulations on receiving this milestone award.
Thank you so much. Thank you.